So the next amazing guest we're bringing on is Peter Hirst. Now, I've known Peter for about probably 7 years now, back when I was working at a little biochar company that was Rechar. But even before that, a few years, like in 2011, when I first heard about biochar from Albert Bates, I was instantly enthralled by the potential for how it can increase the capacity to grow soil and grow better food. And he wrote this book called The Biochar Revolution, which was stunning in showing the scope of how biochar could be produced from a small scale to an extremely large scale. And he really lays out the science of biochar, about why it's so effective and how to complex it and enrich it with compost so that it can be the best growing medium possible. And so Peter is joining us today. He's got an amazing webinar. He's working with Sym Soil to help them create a rich, biologically complete source of compost that uses biochar in addition to the normal compost. So it's going to be really interesting to see what Peter has to share today. And Peter, let me know if I missed anything, but it's exciting seeing what you have to share, and it's really cool bringing you on here.
Thanks for the introduction, Raleigh. Am I on?
Yep, you're on. We can hear you fine. Let me know if everybody can see and hear Brian.
Can you see my screen? Can you see my first slide?
Oh yeah, I can see it. We got a lot of yeses. Yes, yes,
okay.
yes,
all right.
yes.
I will take it then. Thanks.
All right.
Thank you so much for the very generous introduction.
You got it.
Raleigh. Let me give you just a real brief bio of how I got into this. I was going to retire and become the village blacksmith on Cape Cod and started making charcoal as my fuel. Met a farmer named Bob Wells, who some of you may know from New England Biochar, who one day mentioned to me that he knew more farmers than I knew blacksmiths. And he said he saw a little light go on over my head. And so I made yet another transition from my earlier life in the gas and oil business to very briefly to blacksmithing. And then very quickly, I dove in deep into biochar, and in the meantime acquired a permaculture design certificate because somebody mentioned to me, Peter, you know, if you're going to talk about biochar and permaculture, maybe you ought to get your PDC. So I did that, and I got the professional training in holistic planning, and I was called up Elizabeth Pierce one day and just asked her about job opening actually for my son at Sym Soil, and happened to ask her in the course of the conversation, have you considered using biochar? To which the response was, funny you should ask. Long story short, here I am developing biochar products with Sym Soil. So with that, I will jump into what is biochar, and it's basically just charcoal. It's a very special charcoal, as we say. It's produced in a particular way with a particular quality. But if anybody asks you about what biochar is, I would say it's charcoal that's suitable for use as a soil amendment. It does a lot of things, but it really is only one thing. It's mostly carbon. It's got some oxidation. It's got some carboxyl groups. It's got some interesting chemical things attached to it. But it's basically pure black carbon. And one of the wonderful features of pure black carbon is its adsorptivity. You may know it from filters. Activated carbon, which is an even more intensely manufactured form of charcoal than biochar. Activated carbon is used in water filters for aquariums and for your kitchen sink sometimes, and in HEPA masks that some of you may have been using recently, because it's highly adsorptive, which means that it tends to attract and hold organic compounds and biology. And so the same activity that makes it very effective as a filter in air and water is what makes it effective in the soil. It's all these highly porous carbon structures which are bound together with what we call benzene rings or aromatic rings, or little hexagonal honeycomb of carbon molecules with electrons dancing along them, playing musical chairs. And the electrons then become available for all the exchanges that Brian told you about recently. All of the chemical changes, all of the trading of exudates and nutrients throughout the soil, those are all based on the exchange of electrons, not just in cation exchange capacity, which you'll see here in the middle of the screen, but just about every energy and nutrient exchange that goes on in the soil is electrons being traded back and forth. So electrons become the dollar or the Bitcoin of all of these nutrient transaction and energy transactions in the soil. And the carbon in the soil, especially the black carbon in the form of biochar, becomes a virtually inexhaustible bank of electrons to be traded with, to give up electrons for some exchanges, to receive electrons for other exchanges. As a result of this, biochar becomes sort of the WD-40 of the soil. It doesn't change in its chemical form very much, not significantly. It lasts essentially forever in the soil and acts as a catalyst facilitating all these exchanges. And because it participates in so many different kinds of exchanges, I call it the penguin's thermos. You may know the story of the penguin who saw his buddy with a thermos one day, asked him what it was. His buddy says, well, it's a thermos. It keeps my hot stuff hot, my cold stuff cold. The next day, the penguin comes to work with his own thermos, and his buddy says, hey, I see you got a thermos. Penguin says, yeah, it keeps my hot stuff hot, my cold stuff cold. His buddy says, that's terrific. What are you having for lunch today? The penguin says, vegetable soup and vanilla ice cream. In most technologies and with most tools that we use in nature, in mechanics, in just about any form of enterprise, there are very few tools that do a lot of things, a lot of different things well. Biochar happens to be one of those tools. So what we're going to be looking at today is some of the things that biochar does, why it does it, how it does it, and I'm going to give you 2 or 3 things that you can do immediately to start using this tool in your soil. So here's biochar in the form that we'll normally put it in the soil, at least the physical form. Essentially granulated small pieces of ground up charcoal. Here's what it does in the soil. On the left, we have a typical tropical ferrosol, not very fertile, not very productive. And above it, over here on your left, is about the kind of food crop that can be produced in a typical weathered, degraded tropical ferrosol. On the right, we have what's called terra preta, which is the soil that was developed over thousands of years by the indigenous people of South America. Now you'll notice the color of the terra preta is very dark. Terra preta, in fact, is Portuguese for dark soil. This soil here is about 8% on a dry mass basis charcoal, but it's not dark because of the charcoal. It's dark because of the work it has done for hundreds and hundreds of years, building up soil organic matter. You add 8% charcoal to white sand, it's not going to change the color of it very much. But if you let that biochar work in the soil for a relatively short period of time, it'll last for hundreds of years, but it doesn't take hundreds of years to build up. So the dark soil that you see in the terra preta is a result of the buildup of the soil organic matter. And yes, that's a 1 meter stick in both of these pits, measuring approximately 5 or 6 feet of good, rich topsoil. A couple of things to notice on this particular slide. Over here where you see the biochar adhering to the root structure, that's a function of accumulation of glomalin, a protein exuded by plant roots and fixed by biochar in the soil, which can contain up to 15 or 20% of the total carbon in the soil organic matter. On the right, we see my friend Tom Miles standing out in a cornfield in Ames, Iowa a few years ago with the corn he's standing in being treated with biochar. On the left, the untreated soil, and an example of that in individual corn stalks up above. Here's what biochar does in the short-term carbon cycle, that is the biological carbon cycle. If we start with natural carbon in its natural form as biomass, all kinds of biomass comes out of the forest on a natural basis. Much of it today is still making its way to landfills. We interrupt it from its being disposed of and wasted in landfills and conduct a process called pyrolysis, which is just breaking it apart into its constituent elements with heat. One set of those elements can go to make fuel, to make energy, to replace hydrocarbons, petrochemical, fossil fuels. And about 50% of the carbon from any given mass of biomass, we can turn into biochar, return to the soil. So in effect, in working in a partnership with the living trees, we are mining CO2 out of the atmosphere and putting it back in the soil where it belongs. Sym Soil has been mentioned as one of the biologically robust, biologically complete composts on the market today. And in addition to the soil food web as developed by Dr. Elaine Ingham, we have for the first time added biochar and come up with a spectacularly robust product in which the biochar lubricates and facilitates all of the other exchanges you see going on in the soil food web. All right. Now this is the most important, to me, the most important thing ever said about biochar. This is a finding as a result of a vineyard study done some years ago in which Hans-Peter Schmidt, a vineyard consultant and biochar expert from Europe, found that his results, very complex results in the vineyards where some nutrients were increased, others were decreased, but optimized in the nutrient and sugar profile of the wine grapes he was producing that year, confirming literature findings that biotreatments shift plant nutrient uptakes to their particular optimum ranges. It's since been discovered that not only does biochar optimize the nutrient exchanges between the soil microbiome and the plant root, but it optimizes the changes, the nutrient and energy exchanges among the microbes in the soil itself. And that becomes very important a little bit later when we talk about conditioning biochar for application to plants versus the use of biochar in the microbiological processes before the plant is exposed to it. Here's a microscopic view of what biochar looks like at the cellular level when it is raw, right out of the fire or right out of the kiln. What you're looking at is the bare surface of the carbon structure. Here's biochar after it's been conditioned. You may have heard the term inoculated or activated. For reasons I'll get into later, we don't use those terms anymore. This is a biochar that has been interacting with the soil microbiome for a while and built up its own coating, its own varnish of soil organic matter. And it actually conducts its exchanges not only directly off the surface of the raw char, but through this varnish of soil organic matter. Here's one of the great strengths of biochar in supporting mycorrhizal fungi. In this case, you actually see the microscopic hyphal structure thriving on the surface of the biochar itself. Here's another microscopic view of what a good, robust compost will look like on the slide. This is before, the slide was taken before I had a chance to get it and relabel it where it says organic materials up here. That's a big chunk of biochar. And for those of you that are familiar with microscopy, this is a much diluted view of what's going on in the soil. In order to take a microscope slide like this, we mix the solid material, the compost of the soil, with water, and then take a drop of that water. So this is sort of an exploded view of what's going on in the soil. You can see thousands of microorganisms represented here, and bits of biochar floating throughout it, which are facilitating all the exchanges that are going on among these hundreds and thousands of microorganisms. So the first thing I want to turn you on to today is how to make biochar. And for those of you who have ever lit a wooden match and let it burn down and scorch your fingertip, you've already made biochar. Congratulations, you already know everything you need to know. This is pyrolysis right here. This is the heat of the flame vaporizing the solid material in the wood, leaving behind it the black carbon. When you burn a match, that's all it's doing. It's heating up the lignin and cellulose in the match, which are complex carbohydrates. They're very much like sugars, but they're much more complicated and a much longer chemical chains. So they provide structure to the wood, particularly the lignin. Lignin is the plant material that makes the difference between woody and non-woody plants. Those are all carbon, hydrogen, and oxygen, with some other mineral material, but overwhelmingly carbon, hydrogen, oxygen. When they're heated up, the hydrogen and the oxygen are vaporized. They form the flame. The flame then radiates back onto the stick, which continues the pyrolysis process. So you get about 50% of the carbon going off in combustion products and 50% being left behind as char. So I call this the world's smallest biochar reactor because that's in fact what it is. It works very efficiently. Every other process we have for making char is simply an attempt to duplicate what you've done hundreds of times every time you light a match. Here is that blown up a little bit, turned into a more vertical structure of a wood pile, but the same process is going on. You have an initial ignition, which heats up a little bit of wood, turns into a little more flame, heats up a little more wood, turns into a little more flame, goes off in what we call a virtual flare, which is a very, very clean flame, leaving behind the protected charcoal. We call that protected because it has this gas layer around it. It's very, very hot gas. But all of the oxygen in the surrounding atmosphere is being used up in the combustion of this gas. So it doesn't get to the surface of the charcoal, and the charcoal is preserved. And this is what it looks like on a practical scale. This is a forest habitat restoration project in Southern Oregon a few years ago, which we used as a laboratory for using the wooden match pyrolysis process on a much larger scale to turn all these hand piles, which were going to be burned conventionally. They were going to be turned into smoke and ash. And instead, we turned them into a relatively clean flame and a nice pile of char. Now you can see a little smoke being produced at the bottom of the flame here, but you'll notice that by the time it gets up here, all of this smoke has been consumed by the flame. So yes, it puts some CO2 into the atmosphere, but it reduces the particulate emissions into the atmosphere by about 90 or 95% while it's doing it. And it only puts about half the carbon into the atmosphere that would be done in a conventional burn, which would put all of the carbon from that biomass into the atmosphere. One of the most famous uses of this technology is at the Jack Daniel Still in Lynchburg, Kentucky. This is known as the Lincoln County method, where they make filter grade char using the same open burn method. They stack it up, pour about a gallon of 150 proof raw whiskey on the top, light it at the top, and burning from top to bottom, it burns completely clean. You notice there is no smoke being collected under this hood here. That's clean flame and heat, which then is collected and used in the distillery. This is the application in an agricultural context. This is a couple of acres of old grapevines that have been torn out and stacked and dried, and we burned this. This was done on a windy day. You can see the angle that the flame is going up at, Well, we light this pile a little bit toward the downwind side. It burns back into the pile. The resulting flame is just as clean as if there were no wind at all on this particular day. In the middle of this burn, the local fire chief showed up, parked his truck, left his door open, and his jaw on the ground at how clean this was. He saw the heat column from this fire about a mile away and could not believe that there wasn't a raging fire and that there was no smoke involved with this. Now, you can do this yourself. I promised Eric I would cover this. You can do this yourself at home with any burn that you're currently doing in your fireplace, in your backyard fire pit, in your chimenea, in your Weber grill. If you just stack the wood in such a way that it is very light, very airy, lots of air can get to the pile, and light it on the top, it'll burn from top to bottom, preserve much of the wood in the form of char, and then you simply put it out, quench it with water or air quench it. This is the easiest way to make a transition from a dirty agricultural burn to a clean conservation burn. All of these links to this technology, by the way, we are going to make available after this webcast. This one is called the Toucan 1 gallon stove. It's made out of a 1 gallon paint can, a number 10 tin can, and a number 5 juice can, which I have some examples of when we're done with the slides. I'll show you some of these things later. This is a link down here at the bottom where you can get simple instructions to make this online. This is what we call the top lit updraft gasifier. Very much like the open burn, it's lit up on the top. It burns from top to bottom. Burns with a very clean flame. The heat from the flame can be collected and applied to any number of domestic uses. Here's a couple of them at CHAB camp. CHAB stands for Combined Heat and Biochar. Camps that have been held all over the country. People get together, bang things out of tin cans, burn them, make a lot of char, cook a lot of food, and generally have a great time. This is my backpacking stove. This is made out of a 1 quart can, 1 QT. So this is the QT backpacking stove. It weighs about 6 ounces. This is all I have to carry into the backcountry. No chemical fuels, no petrochemicals. Do all my burning in the backcountry with a tiny, tiny amount of pine cones. Very, very little biomass consumed. Much more conservation minded than a wood fire. Now here's the second thing I want to impart to you today is how to mix it, how to use it. This is our chief scientist, Katie Hinson, with a pile of old age composted wood chips, which are developing a very nice mycelial structure. You can see these are highly fungal. After 5, 10, who knows how many years this pile of wood chips has been sitting here, developing this beautiful fungal structure, which we can actually harvest and use in our soil. Here it is with biochar added to it. Up there at the top, you can see some biochar. And here is a culture of all biochar with just a little bit of fungal food added to it. And what took 5 to 10 years of composting and development naturally, Katie has been able to accomplish in a matter of days. I think she said this particular culture that we took this picture from took off in 2 or 3 days and developed this beautiful, healthy, white mycelial structure with just a little bit of food and a lot of really good biochar in a couple of days. The one way to develop it is to mix your biochar with a little bit of fungal food, and you can inoculate it. If Hugh comes back with us, he may be talking later. He's our fungal expert about how to get a good fungal culture started. Biochar is one way to enhance that. Here I am using it in a KNF, Korean Natural Farming trap, where we mix it, biochar, with partially cooked rice. We bury it in some really good soil we found where we'd like to harvest the biology for a couple of days, pull it out a few days later, and we will have harvested good, healthy, robust, biologically diverse, wild soil microbiome for uh, transporting and replanting back into our agricultural processes. Here's biochar being combined with robust compost on a commercial basis. At the top left, you can see the 2-yard bulk bags of biochar with, this is commercially produced organic compost out in Fairfield. This was our first Fairfield site. You can see Katie hosing it down with plenty of water from the tank over there. We then come along with a compost turner, blend this in, and voila, we have the beginnings of commodity compost charged with the biochar, charged with the fungal inoculants and mother cultures that we use to mix it with, and we are off to the races in making a biologically diverse, robust, living compost. Here's some typical results you get in adding compost that is charged with biochar or co-composted with biochar to relatively poor soil. You get your best plant responses in poor soil because you have the most difference in the plant interactions with the biochar in soil that has never seen biochar that is particularly poor. So instead of spending 10 years to go from poor soil to rich soil, you can do it with the biochar, with a biologically robust compost in a single year, and that's the results you're seeing here. Things we have discovered about biochar along the way, which I call black swans and chimeras. One has to do with moisture measurement. If you recall this slide up here, we're putting an awful lot of water onto that compost. In this batch, we learned that the biochar will hold moisture so well, retain moisture and conserve moisture. Not only does it reduce the amount of water that you need to grow your plants, but you need less water than you might think to keep your compost moist. This knocks the squeeze test. For those of you who are familiar with Dr. Ingham's preparation of biochar, preparation of compost without biochar, rather, you want to have about a 40% moisture content. You determine that by simply squeezing a handful of it till you can get a drop out of it. If you add significant biochar to your compost and your soil, by the way, you will increase its water retention capacity such that you can really no longer use the squeeze test. So what you have to do is get a good feel for what that 40% feels like, and then use that field test rather than the squeeze test on your combined compost and biochar, because it's going to take a lot more water in that mix in order to be able to squeeze a drop out of it. That's a good thing because you know you're retaining that water. It's not leaching out, it's not draining out, and it's not evaporating. It becomes challenging in measuring your moisture content because then you have to either recalibrate your moisture meters or recalibrate the feel of your hand. Plant interactions versus microbe interactions. This has to do with whether or not you need to condition biochar before adding it to whatever mix you're using it in. For the plant interaction, the raw biochar will actually compete with the plants for a good amount of time until it gets charged up and conditioned. That's why you don't want to use raw biochar when it's being applied directly to the plants. Applying in any other setting, in a fungal culture, in the beginning of your compost cycle, at the end of your compost cycle, feeding it to your animals, feeding it to your worm farm, any other biological process, you put biochar in at the beginning raw. And the same characteristic that makes it challenging to a living green plant is the characteristic that makes it so vital to boosting and facilitating those microbe interactions. Because that's, after all, what it's doing when it's getting conditioned to go on the plants. It's interacting with the microbial community until it builds up that varnish. poor soil versus good soil. This is a related characteristic. When you first add your biochar to your poor soil, you will get huge plant response because those plants have never lived in soil as good as it's going to be when you add the biochar and a robust compost. Once you get that soil up to quality, however, the biochar still continues to work with the robust compost in conserving the quality of the soil. So your outputs may plateau after you biochar for a year or two, but your inputs will drop. And as we all know, a dollar saved is worth more than a dollar earned. So nutrients that you don't have to add to your soil are even more valuable than the nutrients that you have been putting into it. So your outputs may plateau, your outputs may not increase, but your inputs will decrease year after year after year, and which will give you effective yield gain. Unintended consequences. Remember the penguin's thermos. This will keep your hot stuff hot and your cold stuff cold, which means it'll boost your microbes and it'll boost your plant response. It will tighten up sandy soil and it will loosen up clay soil. It will promote all of the exchanges in the soil that you do want. But if there is an imbalance in the soil that you don't want, this can also exaggerate the imbalance. We've had batches of char that were beginning to go anaerobic, and we did not know that they were out of balance, and the biochar pushed it over the edge. And very shortly after adding the biochar, we knew that the mixture we were adding it to had been out of balance beforehand, because it went severely anaerobic and it took a while to recover. But if your concoction, whether it's a fungal culture, a worm bed, robust compost, is essentially in balance and headed in the right direction, the biochar will continue to push it in the direction at which it wants to go. So you have to be a little bit careful with it. You don't want to put it into a situation that you have some doubt about, because it's not going to reverse whatever's going on in the soil, it's going to enhance it. The final thing to be aware of in using biochar is that, sort of like your moisture measurement, it will skew your carbon measurement. This is black carbon. This is not an organic compound, and it's not in a salt. So it does not count as inorganic carbon because it's not in a calcium carbonate. It doesn't count as organic carbon because it's not a hydrocarbon or a carbohydrate. It is pure black carbon. What does that mean? That means that the biochar itself does not itself increase your soil organic matter. You may get a test back from a lab that counts your carbon just like all the other carbon in your soil, and you may get a surprisingly high soil organic matter reading back from your lab or whatever test you're doing. You have to discount that for the amount of char, because that char is not chemically active. It works in a very different way from the way the rest of your carbon in the soil works. Also, when you're doing your composting and you are watching your carbon to nitrogen ratio, the black carbon, the biochar carbon, does not count as part of your carbon nitrogen ratio for the same reason. It is not chemically, biologically active. Now, it will build your soil organic matter indirectly because it's boosting everything else you have going on in the soil. And if you're building soil organic matter without the carbon, without the biochar, it's going to do that same process even more strongly, building your soil organic matter with the biochar. So yes, we use the biochar to help boost the processes that increase soil organic matter, but the biochar itself is not counted as soil organic matter. The third thing I want you to think about doing today is networking with respect to biochar. One way to do that locally in the Bay Area, we have what's called the local carbon network run by APL. That's All Power Labs in Berkeley, and we are there. We, when I say we are. Sym Soil is their facilitator. Sym Soil is producing co-products with APL and acting as a facilitator to have those products distributed. This is a system of trading carbon in different forms from different enterprises all around the Bay Area. Don't have time to go into it in detail today, but urge you to look it up because it's a model that can be replicated and it can be even franchised in your area. A very effective way of getting black carbon to people who need more black carbon and organic carbon to people who need more organic carbon. And this is part of that process that's used by the Soil Carbon Network. This is the only tumbler composter I've ever seen that actually works. You can actually get this tumbler up to thermophilic temperatures because it's insulated. So using this with the carbon boost from the biochar will actually increase your temperatures to thermophilic levels. This tumbler is insulated to maintain those temperatures, and again, available through the local carbon network. Two other things you can do while we're all sequestered and have time on our hands. Two wonderful books about biochar. One of them by my friend Albert Bates, from whom I have my permaculture design certificate. The other, Biochar Revolution, by a number of contributing authors. Between these two books, there's not much more to know about biochar until Albert and I get together and publish our next book. So this is biochar. This is one turnip on biochar, 32 pounds, including the greens. Any questions? And with that, Raleigh, I would like to take it off my screen and just show a couple of toys. So can you talk me through that? Thank you.
You got it. Thanks, Peter. Yeah,
Can you
really,
keep me on the
super.
screen for just a second? I just want to show a couple of props.
Oh, you want me to
Press
share
the
your
time.
screen? Okay. Yeah, I'll stop sharing my screen.
Okay.
So go ahead and share your screen again.
Share my screen or?
Well, if you wanted to share something, you got to share.
No,
your
I
screen.
want to be on. I want to be on camera.
Oh, you're on camera right now. We can still see you.
Oh, okay.
So we
All
can
right.
see. We can see that panel and you.
This is your biochar analysis lab. We have two bioassays that we do with biochar and two field tests. The first bioassay is the worm avoidance test. Just take an ordinary starter tray, ordinary planting tray, fill half of it with your biochar mix, whatever you're testing, your compost or your finished soil. The other half of it you fill with your control, your soil or your compost without the biochar. Put a handful of worms in the center of it. They will vote with their feet, and they will either run from the biochar, or they will be attracted to it, or they will be neutral with respect to it. You count the distribution of worms. Worms are the best biodetectors in nature. If there's anything chemically wrong with your biochar, if it's too high in hydrocarbons, maybe it picked up some metals, maybe it picked up some pesticide residue from the previous owner, the worms will tell you. If the worms are more or less evenly distributed or even favoring the biochar, you've got a good bioassay. The second bioassay, you use the same tray. This is a 50-cell tray. 72 is even better. This is the germination test. Again, you take your biochar concoction and your control without the biochar, and you plant one seed in each cell. Count the ones that have sprouted after a given amount of time. Use any seed that you would use for, say, microgreens, like winter wheat or radishes, wheatgrass, even sunflowers or peas. Something that will sprout fast enough for you to harvest microgreens out of it will sprout fast enough for you to do a good germination test. Then you can compare. If you have depressed germination, and you can use raw char for this, because the raw char is only going to interfere with the bioactivity that goes on once the sprout has matured and is putting out two leaves. To test the raw biochar, you use it raw in the germination test. Third test that it's done, the field test, is the taste test. This is biochar right out of the oven, right out of the kiln, and right out of the fire. Give it the taste test. You should taste nothing. You're not as good as a worm with respect to this, but your taste is still a pretty good indicator. And don't worry, the char is good for you. It's actually a mouth cleanser, and it's good for your digestive system. The second field test is you get a little on your fingers. Get your fingers good and dirty with it, and then see if you can wash it off just with water. If your fingers come clean with nothing but water, no soap involved, that's a good indication you don't have any residual hydrocarbons in it, no oils, no tars, nothing nasty. That's good, clean char. A couple other things I'd like to turn you on to. You may have seen this stuff, Wright's Liquid Smoke. This is a barbecue seasoning, but it is literally liquid smoke. Any of you who are tinkerers, do-it-yourselfers, if you build any kind of condensation device, put it in the smoke stream, the exhaust stream of however you were making biochar, condense out this aqueous liquid. Not only is it barbecue flavoring, but it's even more important, more powerful in any number of applications in your soil. It's everything from a germination enhancer in very, very weak concentration to an organic, degradable, completely benign herbicide if there's some place you want to knock down the grass or knock down the weeds and compost them in the following season. Liquid smoke is the way to do it.
Cool, Peter. Quick
Okay,
check.
Raleigh,
We're on biochar for about an hour, so why don't we move on to Q&A?
Okay, very good.
Okay, awesome, folks. So now is your time. If you had questions that were burning, we have a really awesome panel here to answer your questions. And one person in addition to joining us is Q from Sym Soil. So Peter, why don't you introduce Q here so people can know a little bit about him and his background.
I will. Q is one of our soil scientists. He is vice president of soil science, as a matter of fact, one of the founders of Sym Soil, and has an extensive background in composting traditions, including those of indigenous peoples from around the world. His formal training includes, let me open up the rest of my message here to make sure I get this correct. His formal training includes Dr. Ingham's soil food web, including microscopy, and advanced permaculture design class from Coil Springs Learning Oasis. Regenerative Design Institute. He's also experienced in construction, and he drives a mean tractor.
Okay, nice. Pretty great intro. Thanks for coming on.
Yes,
Peter.
and leads, by the way, he leads the actual production team at our new site at Sassoon Valley in Fairfield.
Cool,
so,
fantastic. Okay, so
we'll be on to Q&A.
Great, yeah. So there's a lot of folks here, which is amazing. I'm so grateful that we hit max capacity. But that means since we have 500 attendees, we may not be able to answer a lot of really individual questions like, oh, this is my farm. I have too many wood chips. Can I turn into biochar? But, we'll be able to group your questions based on the topic. So we're going to go back first to what Brian talked about with the 5 things you can do really quickly to build soil. And so if folks have questions about extract, tea, BioComplete compost, the soil food web, we're going to hit on those questions first, and then we're going to go to Q and Peter about questions about biochar and the soil making process. So why don't we get started with those? So there is a Q&A window there. You don't want to use in the chat window. There's a Q&A feature, and that's how we'll be able to get to your questions. So, Brian, if you want to go to it, you can go ahead and start answering the questions
Sure.
that you would like to answer.
I'll do one, and then Q, I'll pass it over to you, and then Peter, you want to do a biochar after that? We'll just
Yep,
move
be
around that
glad
way.
to.
Okay.
Cool. Sounds good.
I'm going to take one from Steven, and he asked a number of questions. They're all really good questions, so I'll try to kind of go through those quickly. So there's not just one. There's I think about 6. So I'll go through them fast. The first is age of compost impact on microbial mix. So we definitely do see that certain species become more dominant than others over time. We tend to find that up to about 6 months, you got to have the maximum diversity of species in compost. Then after 6 months, it starts to select for other species. Sometimes that's not a bad thing, especially if we're really trying to make a really good fungal pile. Sometimes a little bit more age to it is not necessarily a bad thing. And then you can always mix, newer sources with older sources to get the microbial mix that you're looking for. So there is age does have some impact to the mix. Advantages, disadvantages of teas versus extracts. The big thing for me, I typically use teas for foliar applications. I use extracts for soil drenches and for root zone injections. There was another question there about, can you use a tea to do a soil drench? Sure. If you made tea and you want to do a foliar and then do a soil drench, no problem with doing so. Like I said, you get a little bit of impact from how much it sticks, especially if you have a lot of mulch. You might get a lot of that biology to stick on the surface of the organic matter. But if you have enough liquid, it should still push a good amount of biology down through the soil. And the next question is, do you test your teas or extracts to determine the microbial mix? If yes, then how? Most of us and my colleagues, we use microscopes. That's the tool of choice. And what we're doing is we're doing a biological assessment. So we're counting. We have certain dilutions that we use. We put them under a slide, and we're counting for the bacteria, the fungi, the protozoa, and the nematodes. And based upon that, we can come up with a profile, biological profile of that soil. There are other tools you can use. You can do DNA sequencing, but it's kind of expensive. It also doesn't necessarily tell you the amounts that are present. You can do plate counts, which I'm not a big fan of because the majority of the organisms in the soil don't grow on the plates, so you miss a lot of biology there. So biological assessment using a microscope is the key tool that we use. And yes, if a tea or compost is primarily bacterial dominated, will that be less effective than one with good fungi to bacteria ratio? It depends. The standard ecological answer is always it depends. If you're trying to grow a crop that's an early successional plant, like a brassica, right, a cabbage or a broccoli or cauliflower, they like bacterial dominated soils. So you're going to use a tea or an extract that's bacterially dominant. If you're going to be talking about a vineyard application or an orchard, they like fungal dominated. So you're going to use a tea or an extract that is fungally dominated. If you're growing a mid-successional pasture grass or a vegetable like a tomato and so forth, you're shooting for more of a 1-to-1 ratio, equal amounts of bacteria to fungal biomass in your compost to your extract. The next question was, what about coating seeds with extracts? Yes, I do that all the time. There's a couple things when doing so. If you coat your seed for a long time, it's going to imbibe. And imbibe means it's going to germinate. So if you do soak it and it's going to imbibe, you got to plant that seed right away. And if you're planting by hand, not a big deal. If you can use something like a seed drill, where you don't want seeds to swell and imbibe because that might impact the usefulness or the functioning of the seed drill, you'll soak the seed coat for a very short period of time, no more than 15 minutes, and then dry it down. You'll still get biology that will fill in the nooks and crannies of that seed coat. Even though the seed coat may look smooth, if you look under a microscope, it's very crinkly. It's got all kinds of nooks and crannies. And you'll get biology to stay in there, possibly go dormant, go in a cyst format, and then wake up when the plant wakes up. Also, do you ever companion plant with corn to get diversity of exudates from other plants? Sure, companion planting is great. I've planted watermelon underneath my corn. I've done some winter squashes and things like that. If you have the ability to companion plant, I think it's a good thing. I've noticed good results from that. And then, do you have any friends with ruminants to graze down your corn crop residues? Would ruminants via poop, pee, and saliva add even more microbes to the soil? For sure, but you have to take care of where you're going to add those ruminants to. In my garden beds, they would step on the sides, kind of break down the sides. I don't want to put ruminants in that system. In my orchards, sure. Well, I've had goats before. I don't currently have goats now, but I've run them in my orchard. It's fantastic. And if you're following like holistic management, grazing practices, um, yeah, ruminants are a great thing if you manage them appropriately. Mob grazing is fantastic because it pushes organic matter in the soil. It doesn't have a huge, long-lasting compacting effect, and their wastes can be very good foods for your plants and also for the microbes in the soil. So, Stephen, good questions. It looks like you were paying great attention, so thank you for that. Q, I'll hand it over to you. You want to take a question that you had lined up?
Hey guys, morning there. Yeah, thanks for having us out here and great to share with everybody. And yeah, so we're going through these questions and we're kind of like picking off them, or we're
Yep.
just running down the
Yeah,
line.
if you find
whatever
one
one
that
you
you want
want.
to answer, that's awesome.
Okay. Yeah. I thought we were just gonna go down the thing.
Oh, down a list one by one. Yeah, no
Well,
worries.
the tough thing is, there's like 100 questions right now, so we probably can't get to every single one.
Yeah. So from John Knapp here, what do we use is compost tea? And so some of the things that we'll use. Sorry, my internet's kind of doing a little bump here, but so it's just like when we're growing the fungus and whether you're culturing it in the compost pile or whether you're bringing in the biochar to then be cultured as well, or you're trying to grow out the microorganisms in your tea specifically. It's like chopped baby oats, and we'll use some malted barley meal. We'll put a little bit of the humic acid in there. The fungus really likes the humic acid, the fulvic acids. We particularly use humic and fulvics that were water extracted, because when you use humics and fulvics that were extracted via like very strong acids and bases, they're kind of denatured, you could say. And so the absorbability and like the plant's inclination and likeliness to use it, like highly chemically like processed and has a sharp pH or anything on that. So humic acid, fulvic acids, the rolled oats, the barley, the malted barley, even using some like sprouted seed teas where you'll get the enzyme release, and you'll kind of, in that germination process that happens with the cracking of the germplasm, and that now the seed has 1,000 to 3,000 times more potency and mineral kind of availability, enzyme availability, then those sprouts can then be blended and put into your tea mix to stimulate that enzymatic process. Because if you can appropriately give enzymes, and you can appropriately give carbohydrates, and provide some of those protein sources, then you're basically providing a complete and balanced meal, kind of optimizing that growth potential there. So yeah, those are some of the things that we use. There's many things. Some cornmeal you can use as well, soybean meal. All of those are definitely like stimulating parts of the protein and parts of the carbohydrate need, both in actual like medium and material growth, also in the liquid culturing thereof. So yeah.
Cool. Thanks, Q.
Yeah.
And so, Peter, is there a biochar question that's kind of caught your eye here? Did you like?
I do. I have a couple of them here that are really good. David has asked to define biochar, and I think that's worth doing because the International Biochar Initiative has a very complex definition of biochar having to do with the residue from pyrolysis, et cetera, et cetera. Lots of people say that it's not biochar until it's conditioned with actual biology. There is something to that, but my definition of biochar is simply char that is suitable for use in soil. It doesn't have to be charged to be biochar. As I said earlier, we put raw biochar into a lot of applications, and we put conditioned biochar into other applications. So it's simply good quality, clean charcoal. Another question that comes up is, what about, I mentioned benzene rings, and that triggered a thought on PAHs, polyaromatic hydrocarbons. When I use the term benzene ring or aromatic ring, I'm only talking about the pure carbon structure itself. That hexagonal pure carbon structure is called a benzene ring. It doesn't involve the chemical benzene. It doesn't necessarily involve any hydrogen bonds, and it certainly does not involve polyaromatic hydrocarbons. PAHs are a class of dozens of chemicals, only 4 or 5 of which are of concern in the purity of biochar, and which are actually very difficult to produce. So if you are a producer of large quantities of biochar, yeah, you want to be tested for PAHs. You want to make sure there's nothing going wrong on a large scale with your process. But for backyard biochar, it is a minor consideration, not one that the chemical engineers tell me I should even be concerned with in producing backyard biochar. So biochar itself is not a PAH. Not everything with a benzene ring is a PAH. And of all the PAHs we could produce, only a couple are of concern. Another similar concern is with pH, or acidity, because typical biochar can be very, very high pH, very alkaline. I have used biochar that had a pH of 11.5 or 12. And that's not a problem, and I'll tell you why. Can you put me on the screen for a second, Raleigh, or am I on?
Yeah, you're on the screen right now. We'd see everybody.
Okay, because I'm seeing you, and I'm seeing you highlighted.
Oh yeah, no, but we'd see everybody who's talked, like you talk, and then we see you on screen.
Okay, very good. This is how we test for pH. Plain, clean water. Make sure you're not using Palo Alto drinking water for this, because it has an unusually high pH. So use a distilled water or a bottled water with a neutral pH and a good quantity of biochar, and make yourself a small quantity of a saturated solution of biochar slurry. And then test the pH of that with whatever method you have available to you. It can be a litmus paper, it can be a conductivity test, whatever you have to test pH, go ahead and use it on this. But understand that what I've made here is a saturated solution. So whether this is a very low ash char or very high ash char, it's still going to come up with a saturated solution and maybe a very high pH. So a concentrated solution may have a high pH, but there may be so little ash in the char, and usually is, that is not going to give you a significant liming effect. I have had, as I said, char with very high pH of 11 and 12, but with a very, very low ash content, which then actually applied either in compost or on the field, had not even a detectable liming effect. One way to assure that you don't have a high ash char is that after you build your fire, after you're done making your nice pile of char, quench it with water, and that'll wash much of the ash out of it anyway. So pH is not a huge consideration. I was also asked about the liquid smoke, the pyroligneous acid, and how much to use. I'm going to refer you to the worldwide interwebs on that, because there are so many different concentrations to use for so many different applications. I don't have time to cover it here. But if you buy one of the commercially available products, this is bamboo vinegar called Seek. It's available from Blue Sky Biochar down in Woodland Hills. It will come with instructions of 4, 5, or 6 page pamphlet telling you all the different concentrations for the different applications. Generally, I can I can say a little bit goes a long, long way. Concentration, dilutions of 50 to 1 are very strong. Dilutions of 1,000 to 1 are common. So I'm not, okay. Do I have time for one more biochar question, Rolly?
Yeah, go for it.
Okay, it's on particle size. How important is the size of the biochar material? We normally process biochar on a backyard basis through some kind of leaf mulcher, leaf shredder, chipper shredder, even one of those convertible handheld vacuum leaf blowers. You put it on vacuum mode and suck up a small pile of char. Pretty much whatever you get out of a typical chipper shredder will be good. It's not, unless you've got a very specialized application, it's not critical. If you get something that looks like some of those earlier slides that I showed, something that looks like this, I don't know if you can see the particle size in this jar. This has quite a particle range from about ½ an inch down to kind of a smoky powder.
We can't see it very well, but was it like raisin sized or?
Yeah, it's raisins down to dust.
Yeah, got it.
And up to ½ inch, ½ inch. While I have it, there is one thing I wanted to show you about the porosity. This piece here, which is typical, would be about 1 gram. And that piece there has about 400 square meters of surface area, all wrapped up and exposed in that little tiny piece of char. So that's a pretty big piece, very light. It's much lighter than a raisin. And that's something else you want to look for in char. It should, when it's dry, it should be very, very light, much lighter than the wood chips that it came from.
Cool. Thanks, Peter. How about Brian? Let's open it up to you. Did you see some questions you want to answer?
Yeah, I've got a question here. Let me scroll on up to it, see if I can find it. All right. This one's from Antonio, and I think this is a great question because I think, Antonio, you're trying to connect dots here, which is a great thing. The question is, inoculating compost tea or extract in soil with no plants would be like putting an army of soldiers in a desert without food, question mark. So yeah, so Antonio, you are connecting dots here. You know, plants play an extraordinary role in the life of the soil biology and making sure that soil biology is going to stay around. Now, I would say this: if you put compost tea or extract into soil that had a lot of organic matter, there's going to be food sources there for that biology to consume. Maybe not all the biology, because a lot of biology is going to thrive better with living plants providing exudates, but there's going to be some biology that's going to be in there chomping down and taking down the organic matter. If you're in a really poor agricultural soil where there's almost no organic matter in it at all, it's been completely nuked, you know, as far as chemical and tilling and things like that, and you just put compost or extract in there, most of that biology is either not going to survive or it's going to go dormant, you know, very quick. Now, Mother Nature will always try to fill in bare spaces. So your early successional weeds will drift in, the seeds will start to, you know, populate, and it will start growing and start the successional change, right? It will go from weeds all the way through to old growth if you let it over a long period of time. So again, yeah, you do want to have plants in the system if you possibly can. If you don't have plants, make sure there's organic matter. If you don't have organic matter, then you got to solve those two problems first. Okay, another question came up, and I kind of answered it, but I just want to, I guess, emphasize it some more because I've seen quite a few questions around this. How to be sure our compost is biologically complete? So there's two ways to do this. If you already have compost, you produced your own, or you've already purchased it and you have it, and you want to know what the biology is, you can get it assessed. If you go to soilfoodweb.com, there are a number of certified labs that do this biological assessment where we do this counts and we provide, you know, the data, so you can find somebody who's locally close to you to do that testing. The other thing I would say is ask the compost vendor if you're going to go purchase compost. If you start asking questions about, have you, you know, done a biological assessment? What is the biological profile of your compost? And they just go, huh? You know, biology what? In your what? You know, at that point, I would be kind of going, okay, you don't know, and the chances are you're probably not going to be biologically complete. So I'm going to go look for another vendor. So you ask the vendors, and those that are clued in. Like you go to Sym Soil, they'll talk ad nauseum to you about the biological profile of their compost. If you go to other vendors that don't have it, then I would probably pass and move along.
Yeah,
Okay.
that's a great question, because we mentioned biologically complete compost, and people are like, what is that? Where do I get it? How do I? Because if you go to Home Depot and you smell it, you're like, this smells like really like just a really nasty fart. That's probably not. I guarantee, like, no one's even looked under a microscope with that. So we're gonna have a list that we're gonna provide everyone where you could find biologically complete compost, or at least robust compost, that someone has looked under a microscope and has a report for. And yeah, we'll just try to source as many as we can. There's, a lot is on the West Coast, some is on the East Coast. There is a few producers in Australia. So we'll try to get this out to you as fast as we can.
Well, you did mention something that triggered something for me that I just want to add
Go for it, please.
on the top of it. We all have diagnostic tools too, that aren't microscopes, and that's our noses, right? So as Raleigh was saying, if you smell really bad smells, you open a bag of compost or it gets delivered and the smell is horrific, like it's overpoweringly bad, then more than likely that's gone very anaerobic, and it's not going to be the biology that you want to be in there. The other thing you can do with compost too is you can look at it, use your eyeballs for visual inspection. Good compost should be the color of a 70% chocolate bar. And it's that darker chocolate. It's not black, it's not tan, it's that 70% cacao. That's a good indicator that that compost has been creating fulvic and humic acids, which means you've got probably fungi in there working, and you've got some biology. So use your nose, use your eyes, you do some visual inspections of it first, but the best way is to have it under a microscope and actually do that assessment.
Yeah,
okay.
Awesome. Thanks for answering that.
Yep.
So
Cool.
Q, did you find some questions that you thought were pretty good? Or do you want us to pick some
Yeah,
out?
I did. A couple things
cool.
that kind of sequence into the next and that. Into the question from Gabriella, mulching question: is it okay to use old leaf debris in place of the wood chips for the brown content for mulching or sheet mulching? And I would say yes. However, there is something that's called a gray mold, and it's in the Oomyces family. And so if you have or notice that on your leaves, and the leaves I feel like especially can house a lot of that material, because all of the leaves like a little cup, and then the water just sits in the little cup, and then it gets some spores in there, and then you got a bunch of them and they mat really tight. And then kind of just depending on your environmental conditions, moisture content, and if that Oomyces was present in the area, then you could potentially get some kind of nasty gray mold. And I have heard of people totally having Oomyces run havoc on their gardens. So, just to caution when you're using some of the stuff that how we said you use your internal investigation system, basically your nose and your eyes, to note that it doesn't smell bad and that it's not covered in a kind of a gnarly gray mold, because that material probably needs to be processed a little bit more thermophilically. It's the way we sterilize a lot of things. But also when we step into the Korean natural farming realm, which there was another question on that, it's a really beautiful thing, because they're not so much, it's not so much focused on the sterilization process as more so on building the probiotic colony content and kind of prepping the environment and prepping the situation so that probiotic life can dominate and can basically hold the ground for growth. And in holding the ground and in the space for probiotic life, there's things that we can use like the labs, live lactic acid bacteria, right? Called LAB, and that comes from lactobacillus, and lactobacillus has a co-evolutionary history and pathway that happened with lactating mammals, and is a probiotic that helps plants, and it helps the gastrointestinal tract of animals. And so we can actually use one of the KNF practices to get that probiotic solution, to get that lactic acid bacteria kind of colonized and into a liquid culture that we can then spray on using, you know, in a foliar way, in a root drench, much as the same way as you would use a compost tea. But we are kind of collecting the like local wild lactic acid kind of bacteria that is in the you know, kind of in your environment. And it's the beautiful thing I really like about that is that it is you know, it's something that doesn't require a pump, and it's something that doesn't require like a mechanized system, which I have a lot of respect for, because that is like pretty ancient and traditional, and like unless you had a running river or a waterfall, or you're sitting there hand stirring it, like most things did happen in a you know, more of an anaerobic kind of an environment. And when I was in Peru, that was, they would make their like bioferments, and bioferments had urine, and they had milk from the you know, from their llamas, and different like plants and herbs in them. And you know, it was definitely it's a way to solubilize like nutrients and minerals. And really like part of the name of the game is like, how do you micronize a mineral and get it small enough to where it can then be absorbed through the root hair, which the root hair will take in the minerals like nutrient by nutrient, right? So it's micronization of mineral, and then it goes to like filling the environment with, you know, nice organisms, right? Like surround yourself with like healthy, productive, happy people, and more often than not, like that is what's going to be in your environment, right? But if you have a bunch of scoundrels and liars and thieves around you, more often than not, you're going to be like losing a lot of stuff all the time, and it's going to be pretty difficult. So that's like probiotic, right? Setting the stage, providing the environment, supporting the environment, and you know, working with those both aerobically, anaerobically is a super good thing. And Korea Natural Farming does some really cool. One of these questions was asking about gathering microbes. Would it be the same if you did biochar as if you didn't? And yes, because you know, it's in the indigenous microorganism collection, which is like using a cooked rice, which is basically a sterilized medium, and then allowing the local cultures, you know, the bacteria and the fungi and different microorganisms to colonize that rice. And then when you put the biochar in there, it's just an accelerator, and it's a stimulator, right, of that pathway and of that process that's going to happen anyways. And then from there, it goes through a series of other processes, and it actually does go through a slight heating process. Although they don't say that it needs to go up into the 160, 140 range. So there's definitely, you know, different trains of thoughts and different ways of building and different, you know, styles of living. But I feel like all totally valid, as long as, you know, we are working on the micronization of things. We're working on the probiotic, like health and wellness and development of things. And as long as we're setting up the environment, meaning the structure of the soil, right? And as long as that structure has the dendritic root patterns that filter and funnel in water and moisture, and they are little micro channels for organic matter, and as long as there's sugars being released into the soil structure that come from the free energy from the photosynthesis process that stimulate the microbes, like, and as long as that soil is covered, and as long as it has like a skin, like all creatures have a skin, they need a membrane to keep them safe from, you know, elemental, you know, things that are happening. Then you know, that's kind of the golden, you know, it's the golden arrow that will get you there, but it's got a lot of tentacles, you could say. So
Awesome. Thanks, kid.
yeah.
Peter, what about you? See some other good ones? Here's a pretty fast biochar one from James. Most plants require slight acidity. He says biochar raises pH. How do we balance these?
Okay, that's a good question. I did address pH, but it's worth coming back to for emphasis. And that is that although the biochar itself has a very high pH, we measure that with a saturated solution, so that biochar that is high ash and biochar that is low ash can both show a high pH, right? The pH itself is not an indicator of the liming effect. The ash content is a much stronger indication of what the liming effect is going to be. I mentioned before, I've used biochar very effectively at a pH of as much as 11.5 or 12 and no problem, because it was so low ash. If you're making it at home, you can wash the ash out of your biochar when you quench it. If you have enough ash in your material that you can see a lot of gray or white, then you may have enough ash to be concerned about the liming effect. If you can't see it, or if you've washed it out, even if you show a high pH, it's not likely to have a significant liming effect at all, because ash is typically a very, very low percentage of good finished char.
Cool. Thanks for the answer, Peter. Now, Brian, let's go. I'll let you do a few here, but I see one that's pretty good. It's pretty burning questions from Johan. He says, I asked about how to protect compost once it's delivered. He says, because I have 5 cubic yards of supposed to be biocomplete compost delivered to our lot, but I don't know how to protect it or take care of it or maintain it. How do I do that?
That is so funny, because that's the one I was going to choose.
Oh, nice. Perfect.
Great minds think alike. So, Johan, it's a great question. Think of it like this. Your compost, if it's biologically complete, it's a living entity, essentially, or it has a lot of living entities inside of it. So you want to be able to protect it from drying down, and you want to make sure that it stays aerobic. Okay, so a couple things there. There's a simple moisture test, and there's the exception that Peter mentioned around if you have a lot of biochar in your compost. But let's say you don't have biochar in your compost. You're going to take your hand and you're going to take that compost, dig into the pile, and you're going to pull that compost out, and you're going to squeeze it with your fist as hard as you can. And what you're shooting for is, as you're squeezing, just a thread of water would be coming out in between your fingers. If you have no water and you squeeze as hard as you can, and you open it and it doesn't really clump together, it kind of falls apart, your compost is definitely way too dry. If you reach into your compost pile and you pull it up and you just start to squeeze it and you see drops of water falling off, then your compost is way too wet. You want to try to manage it at about 50% moisture. So you're going to squeeze and hold to get that thread of water. So moisture management is going to be key. A couple ways you can do this, like here on the homestead, I use a macro bin to store my compost. In a macro bin, they come in different sizes and shapes, but they're typically used in fruit picking or vegetable picking. It's a way to be able to pick a bunch of material and store it. And it usually has little slats on the sides of the bins to allow for some airflow. But it's a good way I can contain it, and I can cover it, and I can store it usually in a shaded spot. I try to keep it there just to not have the sun solarize it and really, kind of bake down the compost. So manage your moisture is a key thing, and don't cover it with an impermeable surface so that it can't breathe either. If you're going to use a tarp, make sure that you at least leave, if it's a large windrow, you said you had, I think, 5 cubic yards. So you probably have maybe in a windrow or pile. Make sure that the bottom third is exposed and open so you can get airflow in. And I probably put some holes across the top so you can allow for some exchange of gases. You can also use shade cloth or some, permeable materials to cover it if you want to try to help capture and keep some of the moisture in there without it drying off. And moisture management is also your climate dependent. If you're in an extraordinarily humid environment, then you may have to protect from moisture getting into the pile. If you're in an extraordinarily dry environment, you have to try to protect it from evaporating off the pile. So just use different techniques and try to manage your moisture, again, at 50% moisture, and you should be okay.
Sweet. Thanks, Brian.
Yep.
Is there another one you want to answer there, or should we turn it over to Q?
I think Q, I could go for it.
All right, Q, did you find one that you felt was pretty good? I see there's a few early ones I was trying to find about like fungal inoculation of piles, but I can't quite see that. So I'll let you just find one that you'd like to answer and go for it. Oh, he's muted. Let me unmute you there. There we go.
One here that I have from Lindsay, and we've talked a little bit about some of this stuff, but just kind of to go over with it. I'll go to the end of her question because I think the rest of them have been answered. And it's just composting in extremely cold weather. So with that, you can basically, you can cut into a hill, and you could kind of make a bay or kind of like a dugout kind of an area to protect it and give you some thermal mass kind of buffering. We also did, when it was freezing in the Northern California area over the winter time, we made bays. We made 3-walled bays out of straw bales that you can kind of stack and like Lego block them up, and then put a big kind of blankie on the top of it and have some wood on them and have a blankie, so you're kind of keeping in all your temperature and all your heat. And even where we were at, there was like, it was freezing conditions, and you're totally able to get that processing to work and everything. And so it goes, just making that right environment and using insulation and using thermal mass, and having a skin on the organism as well, is definitely super important. And grab another one here. Okay, so thoughts on using organic straw as mulch versus wood chips. And wood chips are denser carbon. They're going to last longer. They're not going to get blown away by the wind if you're in that kind of an environment. And, they're going to stimulate longer term fungal growth. Whereas organic straw is, and also wood chips are heavy and maybe more difficult to acquire, right? To where organic straw, you could get a truckload of them in, you could bring in a bale in the back of a little teeny car. It's pretty efficient. It spreads out pretty far, and it is a carbon source, but it's also like a great kind of an insulation source. So where the straw might insulate you better from evaporation, if you're in a high evaporation area, and but it's not going to last as long as the wood chips, So those are kind of like a couple different ones there, and a couple different options that you can do. And just on there's somebody asked just about KNF, and there's a KNF Hawaii, and that's where I took my training out there with Drake over the winter time, and really awesome work that he's doing out there. And yep. And then I'm looking into another one here. I think I got. So vermicompost is material resulting from vermicompost. How does one finish from the compost from a worm bin? So using vermicompost is going to be, it's super awesome because it's another spoke of the wheel, and it's great because the worms are basically like squishing and squeezing the microorganisms, and that's a really great thing, because you're getting the enzymes from the worm's digestive systems, you're getting the nutrients that have already been micronized, that have already been coated with acids and enzymes that are coming out of the worm's digestive tract. And so it gives you another like broader, diverse spectrum of minerals and nutrients and enzymes. And you're going to use those worm castings much as you would use compost itself in compost tea, and you would liquid extract those in your bag. You could even just put it in a simple bucket and mix it up. Hey, excuse me here, sorry. You can put it in a bucket and mix it with the worm castings, and then have that liquid and pour it in your pots. It just depends on what scale you're working off of, and it depends on the volume of product that you have to work with, and how far you're trying to get it out. Like with Sym Soil, we have these really awesome extractors, and it's in a 250 gallon square ICB tote, and it basically, we have a stainless steel shaft that goes into the tea bag, and it blasts it from the inside out. So your extraction rates are really high and they're really fast, and you can get like your water will go dark chocolate brown in 15 minutes, and other compost processes like that takes a while to get that high of an extract out. So it depends on your budget and your equipment, and like how how valuable is this crop to you? And like, yeah, we all know food is priceless, and it's becoming more and more aware as like supply chains and things are kind of closed off into certain areas. We're like, yeah, it's super important. But I mean, also, if that's how you're making your livelihood, then that whatever your agricultural product is, then yeah, you'd want to step up your quality as well, and your mineral potency, and it's one thing that we talk to a lot of farmers about, is that the beauty of the complexity of the soil food web and the beauty of the complexity of the humic acids and the minerals is that those complex proteins, those complex acids, those complex minerals and molecules that have been combined and um, put together, pushed together, um, they are going to make complex flavors, and they are going to make complex cannabinoids, and they are going to make complex proteins. And so the plant really will be able to express its full genetic growth and potential. And I believe, growing up, with natural medicine and natural food, is that if you have that mineral, availability for a developing brain, or if you have that mineral availability for a developing animal that you're working with, that you will have a more robust and a stronger and a healthier and a more integrous, animal, plant, human. So that's why, like, this whole integrated system, which is like the age-old system of the forest, it is the way of the forest and the earth, is like really truly being able to be understood as like, hey, these are our solubilization processes, and we're going to industrialize them so that they can reach further out, and we can kind of like hybridize the technology, because it's kind of hybridize or die as far as like evolution goes. So we'd like to ride the wave as well as we can with all that.
Awesome. Thanks, Keith. That's another topic I wish we had a ton of time to
Yeah.
talk about, how the health of your soil becomes the health of your body. Like, they are so interlinked that a gut imbalance can also be correlated to a soil imbalance. Like, if the food you eat is filled with Roundup and other toxic pesticides and herbicides, and you're absorbing that in your gut and through your food, you're going to be dealing with some nasty diseases down the line. But so here was a general gardening question I had, I think, before we get into another biochar question. It's from Alexander. He says, the majority of organic waste on my allotment is either tree or shrub cuttings or weed rhizomes. How would you go about composting these when you can't invest the time in a thermophilic composting or turning? This could even be a Hugelbed question.
Sure. Yeah, there's a lot of uses you can take with that material. So compost. There's kind of three ways or way we process or make compost. There is the thermophilic process, which is really a process of time, moisture, and temperature, and you're managing those three things very specifically. So you have a recipe that you build. You have a certain percentage of high nitrogen material, a certain percentage of green material, and a certain percentage of woody material, and you intensively manage it in a very short period of time. It heats up, it gets to a certain temperature that kills off pathogens and weed seeds and all of that kind of goodness. And then you usually end up with a really good biologically complete compost if you did the process right. But it takes some practice and some skill and some knowledge to be able to do that. And I would suggest anybody who wants to, for sure, learn how to make thermophilic compost. It's a fantastic thing to do. But if you don't have the time or the expertise to do that yet, you can make static piles. Static piles will not kill pathogens, and they won't kill weed seeds. So you have to be okay with that. But a static pile, you can get the right mix of, again, greens and browns into that pile and let it compost over, let's say, a year period of time. So it just takes a lot longer to make good compost, to kind of have it break down. So that woody material you were talking about, you could use that in a static compost pile. Raleigh mentioned hugelkultur. That's something I use on my property quite a bit. I like to take that wood and actually put it into hugel trenches. Where I live, it's very dry, so making hugel mounds is not so good. They dry out too easily. I like to bury my wood in the ground and make hugel trenches, and so that's another avenue you can use for it. And if you were able to chip it up and, again, you aged it out, you could use it as a mulch. Again, it's going to put organic matter on top of your beds, provide that protective layer. So I think you have a lot of options to be able to work with that material. Anybody else
Awesome.
want to
Thanks,
add?
Brian.
Yeah, I
Peter's
think
got
Peter wanted
something
to
to
add.
add.
Yeah. Oh, big biochar out of
Yeah.
it.
Yeah, that was actually a great segue, because another intermediate possibility is biochar. If it's small, people ask us, well, I got a pile of wood chips, can I make biochar? Rather not try to make biochar out of wood chips, because that's where you need some technology, and it gets difficult. Other people say, I've got big logs, should I make biochar out of it? Well, big logs are tough to make biochar out of. Well, but there's an intermediate size between, say, 1/2 inch and 4-inch diameter that's perfect for biochar. So if it's really small, compost it. If it's really big, hugelkultur, and if it's in the middle, biochar. Nothing should go to waste.
Agree. Yeah.
I had a couple other questions I'd like to address quickly too. I get a lot of questions about ash. Is my ash as good as biochar? Ash is very, very different from biochar. Ash is the highly alkaline mineral residue from the plants, has very, very little carbon left in it. It can be useful for making soap. It can be useful for liming if you really need to raise your pH, which is unusual in many places. But it is very different. It should not be confused with biochar. Now, you can separate the two. If you get a very high ash residue from a wood stove, for instance, it's got some charcoal in it, you can run it through a sieve and save the big black chunks for charcoal, and separate the small gray stuff and use it for whatever use you may have for ash. But they are very, very different. The other question I get is application rates, and there's probably a whole day session we could do just on application rates. But a really good rule of thumb is that if you're just starting out to improve your soil, if it hasn't had either biochar or compost yet, a 50-50 mix is really good for a lot of reasons. The char will complement the compost. The compost will condition the char to get it to go in the soil. And for a first application, what we've calculated for some of our Sym Soil products, a 50-50 mix is a perfect match between a really good application rate for the char and a good application rate for the Sym Soil itself. So Sym Soil has a 50-50 mix based on that. Back to you.
Apparently, Johan says he used a compost called Cold Creek Compost, and he says that got biologically assayed. So yeah, send us the document if it's verified that it has the right ratio, and if it's verified the Soil Food Web people, then it could be listed. So please,
Q might know
Johan.
about that Cold Creek off the top of his head.
Oh, wait, is that, do you guys know?
So what?
That's
Wait,
what
what was the question about?
it says, Johan says the compost I bought is Cold Creek Compost. I just checked it, and it does have a lot of lab tests of nutrients in the biological assay as well.
Yeah, so Coal Creek is a product that I do know various organic farmers to have used, and it has a great nitrogen content, and it does have minerals, and it does have nutrients, and we personally, you know, that was one of our workhorses that we were going to kind of integrate into our process. However, for us, because of, and I think it's due to the grease content, because they use a lot of chicken that they have a contract to take chicken waste, and so it has a very, like, you know, a barbecue and or like a kind of a cooked chicken smell to it a little bit, but it was hard to get our fungi to grow on that, and I attribute it to the oil content that was in the material. And so this goes back to the two pieces, right? Like, one of them is that, like, how do you take organic matter and how do you take plant and animal matter, and how do you prime it and process it and get it to become organic matter? And organic matter is something that you have to put into the soil structure, right? It's sand, silt, clay, and organic matter, right? And every time you till your soil, every time you farm in your soil, every time you grow a crop in your soil, part of the organic matter is used up. Okay. And the organic matter is going to be kind of like the structural support it's part of the structural support into the soil network. And if you use up all the organic matter, then the structure collapses, right? And when I say the structure, it's part of the way that the soil aggregates. It's the way that the soil clumps and bonds and binds together. And if you lose the organic matter, it loses part of that capacity, right? So one of the things you have to do with soil is you have to replenish the organic matter. And if we go back to the way of the forest, right, there's always organic matter falling onto the soil. There's always poop dropping onto the soil. There's always bugs and little carcasses and little things falling onto the soil. It's always covered. It's shaded. It has roots growing down into it. It has moisture getting pumped into it. It has sugar getting pumped into it. So one piece of that process is the organic matter process and the part, right, which is very, very important. And if you're on a super budget, right, I would get commodity compost, and I would mix it into the matrix of my soil structure so that I had my organic matter quantity up to par. And then I would take high quality, robust compost, and I would make liquid extract out of it. And I would make compost teas out of it. And I would then use that in a liquid application, right? And because now I've got enough organic matter that can hold enough moisture in the soil, that can kind of keep the structure together, then I'd put those microorganisms into it. Now the microorganisms, they are the machine and the processors, right? So they're like micronizing and chelating and processing, but they're also releasing the glues and the enzymes to help the structure as well. So to maintain structure in a soil, it's a two-part piece. One part is organic matter, and the other part is the byproduct of the function of the microbiology, which is the enzymes, which are the glomulates, which are the glues, which are all of those things that will then also hold that structure, right? So, yeah, it's definitely a two-part piece when you're thinking about like, hey, I want to farm this soil. Because also, like, kind of initially, unless you're dealing with a really well-developed, like, loamy soil, like, you might have to go in there and manage it. Like Brian was saying, you have to go in and manually break it open. Even Elaine says that. I personally had that experience working in a heavy clay soil. If you can break it open and you can get the organic matter in there, and then you can start pumping it with teas, and then you just take care of it in the sense that you mulch it and you keep plants growing on it, whether they're your target crop or whether it's the cover crop, you're still keeping the sugar trade going. And as long as the sugar trade's going and there's organic matter and it doesn't evaporate or get too damaged by the sun, then like the ball keeps rolling, like the bus keeps moving, you know, you keep fueling it and the bus will roll. So, yeah. And somebody also commented about a mineral analysis, and like, you know, there's a lot of different viewpoints on this, but the mineral is the mechanical, right? Like these minerals mechanically function a certain way. And it is good to know what mechanical operating system that you have in your soil. It is not completely necessary, and it doesn't mean that you will not be able to farm, right? It just is another tool that you have that will help you to be a more successful operator. And the biology is kind of the really awesome natural method that, like Brian was saying in his presentation, it's the mining of the mineral, right? It's the immobilization of the mineral into the body of the microorganisms, and then in it, at its death and at its birth, there is the release of the mineral. So then it goes back into its mineral form, you know, and so it's a two-part system. Like, it's not like there is the mechanical function and there is the biological function, you know, and that's just like thus far what we have come to know it to work as, you know.
Look, I'd just like to add on to that. It was fantastic what you said. You know, for me, when I look at soil, I think it has three main properties, which is its chemical properties, its physical properties, and its biological properties. Unfortunately, in today's agricultural system, they focus on two of those things, the physical structure and the chemical, and they completely ignore the biology. The reality is that the biology will change the chemical and the physical structure to what the plant actually needs. So if you focus in on the biology, you'll take care of your chemical and your structural problems. Yeah, it's fortunate. The focus has been in completely the wrong area. Focus on the biology, you're gonna create environments for plants. Yeah.
Well, and it just goes back to the simple, like, this is a circular planet. Like, we are a human race, right? We are all animals, and we are on a circular system that is an integrated closed loop system. And if you do not honor that, like, you will have problems, right? It's just like, there's not really a good way around that, because it's like, yo, closed loop, it's a circle.
Yeah.
Like, roll with it, or be a square and see how, well, you do not roll.
Yep.
I think we're all learning, and we're learning how like complex the soil is and should be without nutrients. I think because the way it has been for a long time is just dumping NPK on it. And if you're a body, you can't exist on just Cheetos. It's like if you're just eating Hostess cupcakes and Cheetos, like you might just live, but you're going to get really sick. Well, maybe,
how do you know that, Walt?
I'm just kidding.
I'm going to do it. I'm going to do the Cheetos Hostess Cupcakes challenge. I'm going to be the strongest. And I'm going to laugh at all of you and say, organic and compost is a lie. I did the Cheeto test.
You sounded like you were speaking from experience there.
Yeah, maybe when I was like 15. Okay, so real quick, folks, we're just about hit our limit here. We're probably going to only go on for like 3 more minutes. Unless, if you guys want to go on longer, that's cool. If the panelists want
I have to actually drop it.
Yeah,
Raleigh, I actually have a question I've been asked to follow up on, and I did not quantify the application rate. I said it's 50-50 compost biochar is a pretty good application. Cool. And I did mention in my presentation that the Terra Preta has a char content of about 8% on a dry mass basis. And again, we could have a 3-hour seminar on how to calculate dry mass percentages. But if you approximately equate volume with weight, of finished wet char with finished moist soil, you can get in the same range of about 3 to 8% by volume. Will be close enough to 3 to 8% by finished weight, and we start to see real results at 3%. They start to taper off after 8%. So that's the sweet spot.
Cool, thanks, Peter. And Brian, if you got to get going, super appreciate your time.
Sure.
Super grateful for having you on, man. Thank you.
Thank you, everybody. And thanks to all my fellow panelists.
Thank you, Brian.
We will say goodbye to Brian.
See you, everybody. Happy farming.
That's right. So I guess like Peter and Q, it's like however long you guys want to stay on, that's totally cool. We can keep answering questions.
Well, I'm seeing we still have over 200 people hanging in.
Yeah, totally. I mean, it really depends on if you guys want to stick on.
A little longer, yeah.
Okay, great. But if we got to go, then we can close it out. But I'm cool to stick around a bit longer, get these questions answered. Well, you just crossed the 2.5 hour mark, everybody. Give yourself a round of applause. 2.5 hours of soil biology learning. That's pretty cool.
Fantastic.
All right. So
thank you to the attendees for hanging in here with us for that.
Yeah, I know. 280.
I shouldn't have said anything. I see the numbers dropping off now.
Dropping like flies.
One thing I answered it, I typed it in, but was actually at a biochar course that I took with Peter some years back, and someone was asking in reference to hardwood versus a softwood, and I don't know if everybody sees the answers that you type or not, but if they don't, then it was interesting that particular day at that particular workshop, there was an engineer there who was very curious about biochar, and he basically was like a bioengineer, and they would do like filtration systems for like chemical like facilities and like highly contaminated zones. And he said what they had been finding with their studies was that hardwood, due to the density of the parenchyma cells, I believe that's what it's called, the initial stacking of the cells as it like pushes outward like this in a plant, like in the plant growth kind of like schematic of how it operates. It basically, they stack cells on cells on cells on cells, and those are kind of the rings that you're seeing. So the harder woods have a denser stacking of those pores, And so then that means that filtration capacity is going to be greater, and its ionic charge, like grabbing potential, bonding potential, is going to be higher. And that they were using those for actual like filtration and for minerals and yeah, and just people eat biochar as well for your, like you were saying that earlier, Peter, like you, like in natural medicine, like when you're sick or you have diarrhea or like your tummy is all messed up because you got a bacteria in it, you'll take activated biochar and it will like bond and it will kind of calm and cool, and it will like basically take that bacteria, house it, and let you pass it and kind of move it on and stuff. So
Yeah.
all pretty good stuff there.
Yeah, what that engineer was talking about was the a filtration application where you're dealing basically with a pure char or with a material that is mostly char. The particle size does make a big difference because it's a mechanical process. It's like having a smaller sieve size in any kind of filter. In the soil, however, it turns out it doesn't seem to make that much of a difference. Some of the most highly absorptive chars made, the most biologically powerful chars, are made from dug fir, which is a softwood. It's among the harder and denser of the softwoods, but it's not as dense as oak or madrone or manzanita, for instance, which makes a very mechanically dense char. Now, when you get into other materials other than wood, something like walnut shell in particular, then the pore size does become a factor in the biological activity in the soil, because a walnut shell is like plastic and it has to be charred at a very, very high temperature to get it to puff up a little bit. But barring walnut shells and material of that, coconut shell is another one that needs to be done at a high temperature to achieve the mechanical porosity. Aside from that, the process seems to be far more important than the wood species.
Definitely.
Here's a really good
so
question. Oh, sorry. Like, okay, this is one for you. I think everybody here. I thought this one was super relevant. It's kind of the purpose of the webinar too. Given the compressed time context of the virus impact, how effective are these treatments in the short term, this growing season?
You want to take that on compost first, Q?
Yeah. And it's funny actually, like looking into like virus histories and everything, and I mean, okay, so it's like in a plant cell, there's mitochondria, right? And mitochondria are believed to have been an external organism originally, right? And then either through the acquisition process via like eating something, right? And then it learns to hybridize and live within the system. And then over multiple, because we don't really know how long it takes for a hybridization process of bacteria and plant to basically into the DNA codex to then have it be part of the systematic function of things, right? Like I believe that happens over quite a long period of time. However, so there is like these adaptions and these hybridizations that happen from what can originally be a pathogen, what can originally be a predator, that might not be able to predate successfully on its prey, and thus then becoming integrated into the system. So like, viruses, like over time, some of that genetic material is written back into the cycle, and it becomes like this. I think it's not quite so, and it takes a while for us to kind of like balance and kind of stable and get to a homeostasis level. But because of the back to the great wheel, right, and like a bicycle wheel with the many spokes, like as we have diversity and as we have strength and integrity within that diversity, many are our spokes, right? And our ability to roll and our ability to withstand external pressure is greater, right? So like the forest is a self-sufficient system because of its diversity, right? And because of how all of the things work together. So like same within the compost pile, because it is diverse, and the more diverse that your compost pile is, it means that your plant becomes more and more able to deal with pathogens, to be able to deal with viruses, because we have it's like if you're friends and you're cool with all your neighbors all around you for miles around you, right? And you could call on anybody you need for help at any time to come and back you up and to help you out and to like give you a hand and to loan you some sugar and to like bring their machete or whatever is going down that you need help with, right? Then you're really capable of surviving, and you're really capable of flourishing, and you're going to have a good time and probably like be healthy and successful and raise healthy kids and at least feel good and be at peace, right? And not get consumed by some other pathogen. And I feel like that's the same with the compost. That's the same with, you know, the diversity that is so important. And even in farming, right, that's why we have multiple different species of cover crops. That's why that UC Davis recommends a minimum of 15 different types of cover crops to successfully provide all of the sugar diversity and mineral diversity and enzyme diversity that the microorganisms are going to want in the soil structure, right? That's why we would put different cover crops for different times of years to provide different types of sugars to be able to grow. But it is the diversity that is the strength and the integrity, right? Like that's like big wheel, right? Many spokes roll strong.
Almost as in life begets life. Huh. But also, and talking about the amount of time that like you're trying to grow as much food as possible in the short term. As Brian showed, huge turnaround can be achieved in one season. You know, it's like springtime. You can get these sources of compost right now, start making extract from it if you can only get a little bit, and you can start using this now. Like start sprouting your seeds or get your starts, get this in the ground right now, and you can start seeing an impact ASAP. Oh, Seth, are you still there? We had a Seth question, but it looks like see if he just bounced. No, Seth just left. Oh, well. Seth
Well, yeah, I've got a comment on the time factor.
Yeah.
Biochar is really in its glory when time is a factor as it is now. We of course we all want to see short-term results. We want 30-day crops, all of that stuff. But biochar works two ways. Not only does it accelerate all the other processes that we have been talking about, can shorter the maturation of thermophilic compost, et cetera, but in itself, it goes to work in the soil almost immediately. It will work if you make biochar the day you put your seeds in the ground. You can put the finished biochar in the soil soon enough to nurture those plants that you have just seeded. You can get an immediate plant response effect from it. The longer term soil building, soil organic matter building takes place over the course of the first season and every subsequent season, but the plant response effects on biochar are virtually immediate.
Sweet. Okay, so Seth had a question about, just a great question on biochar porosity. Says, what factors determine the porosity of biochar? Is it the type of wood? Is it the temperature? Is it the time of burn? You know, how do you make the ideal biochar?
There's several different degrees of porosity, right from what is visible down to the molecular level. There are cavities and pores and openings and crevices for everything from the macro to the molecular level. But generally, at the macro level, the char will maintain the gross morphology of whatever plant it came from. If you're dealing with oak, you can recognize that oak grain in a piece of char that comes from it. So at that level, the porosity, as we talked about earlier, for filtration purposes, can be very important. You want it finer for finer filtration functions. At the molecular level, it's far more a result of the temperature, the residence time, and the amount of oxygen in the pyrolysis environment. It's not so dependent on feedstock. Those pores can get clogged if it's not being produced at a high enough temperature. Those pores will open up in the sweet spot of about 600 to 750 Celsius. After that, you get changes, slight collapses in what's called the amorphous graphene structure, so that the larger surface areas will be reduced a little bit. But the really functional molecular level chemical surfaces and the electrical effect will increase at the higher temperatures. So generally, you don't want anything as dense as walnut shell if you can avoid it, unless you have a very high temperature process. But any kind of, in the usual backyard process, home process, the more common processes, any kind of material, the woodier, the better. Grasses, for instance, have no lignin in them, so they're not going to give you as high a carbon yield. But as far as the porosity itself, it's much more a function, at the important levels we're talking about for chemical processes, much more important that the pyrolysis occur at a sufficiently high temperature than it is what kind of material you're using.
Cool. So Seth, I hope that answers your question.
I have one question came up here by Matthew Kilumans.
Yeah, that was a good one. That was a good. It was a Terra Preta question.
Yeah, well, he was actually asking about the pump types for spraying compost teas and extracts, and I just wanted to just comment quickly on that. And diaphragm pump is best. Maybe a centrifugal pump, but you want to stay away from hydraulic like ram pumps, and you want to stay away from like hydraulic ram piston pumps and impeller pumps, right? Anything that has like a manual like rip, press, crush blade to it is probably not going to be your like your best option for that. And then if you apply compost tea and it sits near the surface, sure, the biota will colonize the rhizosphere as it has been introduced in the soil, if the soil structure is adequate. So not only is it the soil structure, okay, so if you're introducing a tea and you're introducing something, it's not just the soil structure, but it's also the moisture content in the soil, right? And then enough moisture gets out there to allow it to move down into the soil profile and allows it to be able to migrate around. That's another piece that's like super important. So, and I know with the Korean natural farming world, that's why most of those drench, applications of stuff will be done in a wetter time of year, because the water is life and water is mobility, right? And so with the water, they can move. That's pretty important thing there.
Cool. Thanks, Q.
Yeah.
All right. Oh, there's so many good ones here. I think when Peter gets back, we'll save the Terra Preta question for him, because I'm sure he's got that a lot. Say we're going to call it 6 minutes from now, but I think, Peter's back.
I'm back.
So Peter, there was a Terra Preta question here.
Yes.
It's from Matthew. It says, is it possible to make Terra Preta out of situ, or in the terms only given to the type of soil created in the Amazon?
Yeah.
As in situ over a period of time, thereby transforming the structure of the soil?
That's a really good question.
Can you
And by
make Terra
good
Preta?
question, I mean one that's really difficult to answer. There are components in Terra Preta that are not completely understood. In situ Terra Preta, we find, for instance, pottery shards. And there's lots of theory as to what the pottery shards do or where they came from. One speculation is that it is just an all-purpose waste dump and all the broken pots happen to end up there. There are other theories that the pottery shards actually serve some function due to their porosity and additional water holding capabilities. There is a book on the subject called Terra Preta, The Dark Earths of the Amazon by Christoph Steiner. And I highly recommend that book because it's a very complex, very extensive subject. Generally, we can duplicate everything that is most important about Terra Preta to us, which is both plant response, durability, and resilience over time. But whether or not we're actually making terra preta, we don't know yet.
I'm sure with, like with those guys do at Soil Food Web, like Brian, he looks at a Soil Food Web report and they look at it and they verify the, like the biomass, the complexity, the fungal to bacteria ratio. Because if you're making compost that has an amazing fungal to bacteria ratio, then you're making exactly for the type of succession, that could be for a forest or an orchard, or it could be less of a fungal bacteria ratio. So it could be ideal for your row crops, or it could be less than that, and it could be for flowers. It's like you can target the type of plant you want with the right kind of fungal to bacteria ratio and the complexity of the organisms in your compost. Now, is that terra preta? Yeah, it's not terra preta, but you could essentially create something that's like terra preta for these plants.
Well, the terra preta was developed specifically in situ. Authentic terra preta was in the tropical rainforest, so it's going to reflect the geology, the biology, and the microbiology of the area that it was developed in. But there are, I'm sure, analogs to that could be developed for temperate grasslands, boreal forests, and other areas that may not reflect the specific formulation of terra preta, but would serve the analogous function in other zones. Q, did you want to say something about that? One of your superpowers is plant succession and community succession.
Yeah, one more time with that. I was kind of answering a side question here.
So
community succession
from bacterial to fungal, from grassland to forest.
Yeah, well, it's a really beautiful thing actually, because as the enzymes and the acids and the glues and the glomulins are released from the stimulus of the microbiology, then the structure is then enhanced, right? And as the structure is enhanced, then the productivity of the plant is then enhanced, and then it will produce more organic matter and it will have more root matter. And then as the foliage production is higher, then usually the animal consumption and production will be higher off of that natural system. And then you'll be getting more organic matter down into the soil and more available nutrients. And so the structure will build, the percentage of organic matter will build. And as those build, then in that the evolution happens, right? And there's more organic matter and there's more carbonaceous material, so that the fungi can then kind of grow at a greater rate, and then it will kind of shift and changes it. And it also changes the it changes the texture of the soil. So the texture, the structure is changing, the organic matter percentage is changing. And as we know, as every farmer knows, right, whether animals or plants or fungi, that as you change the environment, as you change the climate and the conditions with which your animals are growing, you will see a difference in the growth and the production and the reproduction of your animals or your plants. And so it's like a self-serving, self-evolving system that grows. And then it's amazing, right? And Paul Stamets has talked about this, so that there's a collaboration that happens between, so as the weeds evolve to the grasslands, as they evolve to the bunch grasslands, as they evolve to the bushlands and the vineys, and then it goes to the deciduous forest, and then it goes to the evergreen forest, then the evergreen forest is then climaxed. But it's amazing how it's climaxed, because it's climaxed by, so there's a bear, right? And the bear is eating and breaking into the honeycomb, right? And the bees have a symbiosis that's happening with some of the red polypores, right? And some of the different fungal colonies and species. They have, part of them are like they're in a homeostasis like with each other, right? And they kind of carry some of the same spores and material. And so then the bear breaks into the bee hive and he eats the food, right? And he's an apex predator, right? So we have apex predator, and then we have like a bee's kind of an apex like bug, I would say, or probably a better word for that. Bee is basically managing the fungus to have this like apex caretaker, which is apiary apex, caretaker of the forest system, of the plant system, basically from everything along the whole successional pathway. And then you have a predator of a forest ecology, which is the most evolved plant production system as well. So you have two kind of apex creatures coming together, and then you have fungus, which fungus has been known to be like the co-evolver of like life and creation, and has changed like the whole environment, acids and combine minerals and resources and mobilize them to then kind of support and promote like life, right? So the bear then, after eating the bee's honeycomb, then goes to the tree, right? And he's like scratching on the tree and like rubbing himself on the tree, and part of the spore from the red polypore is then carried by the bear in his nails and in his claws, and then it is then broken to the cell walls of the tree, and then that red polypore then uses the tree to eat it, right? And now we've climaxed our evergreen top evolutional like process and growth cycle of our plant kingdom, of plantae kingdom. And then it brings it back into the meadow, right? It brings it back into the weeds and the grassland so that it can evolve again. And it's just some really beautiful, like, kind of poetry of how, like in the process of the caretaker and of the predator, it then like evolves and resets the stage so that life can kind of do its dance and growth again, and just again echoing how important it is across the board for mycelium and bacteria and diversity and predator and insects and insectivaries and just like that whole kind of network is a really a really functional, healthy, kind of beautiful like dance there.
Nice, thanks, Q. I think that's good as a wrap up. We're almost at the 3-hour mark here, so y'all are champions for sticking around. Oh no, I'm out of focus. Thanks for sticking around for 3 hours here to learn about soil building and biochar. That's so cool of you all. And I'm going to challenge everybody. We're going to send out resources here, and we want you to start doing a trial at any scale you can. We're going to get you the resources for where to get the best compost, how to make extract, even if you can only get a little bit of compost. And we're going to ask you, grow your plants, take pictures, because the more people record they're doing this, it will prove to yourself how effective it is, and it will prove the entire world how effective, creating amazing compost is. And especially since we're all in an emergency, we should start all doing it together. And take a look in your emails. You'll get a replay. Thanks, Q. Thanks, Peter. You guys put on an amazing webinar, and everybody got a lot out of it. So thank you. Thank you all. Thanks for giving us your time.
Thank you, Raleigh. Q, thanks for jumping in on late notice and